Hey guys, it's Miss Adelini and I am back with round 2 of macromolecules, so get geared up. In this lecture, we're going to be talking about both proteins and nucleic acids, the two macromolecules that we did not focus on in our last lecture. Proteins is everyone's least favorite macromolecule, and I don't really blame them because they tend to be very, very complicated. But I'm going to try to break down the different levels of protein structure and talk about how each of those levels contributes to the protein's overall function. So we'll start off with just some general information about proteins. They're made out of carbon, hydrogen, and oxygen, just like our carbohydrates and our lipids. However, they also add in two new elements that we did not see in the carbohydrates and lipids, and those would be nitrogen, N, and a very small amount of sulfur, but those sulfur atoms are only found in certain protein building blocks. Here it says they're used to accomplish all life functions. We often call proteins the workhorses of the cell because they do so many jobs within the cell. Towards the end of our section on proteins, we'll go through a few examples of protein functions, but just know that they are not limited to the functions we'll discuss today. The monomers for proteins are called amino acids, and a lot of people are tempted to say amino acids are the monomers for nucleic acids just because they both have acid in the name. So you'll have to be very careful to remember that amino acids go with proteins. Amino acids are joined together by a special type of covalent bond called a peptide bond in the process of dehydration synthesis. The process of dehydration synthesis between two amino acids is shown here. We see that our amino acid 1 is going to lose an O and an H, a hydroxyl group, and amino acid 2 is going to lose an H, so that we are going to form one water molecule that gets lost. Then a peptide bond, a covalent bond, will be able to form between those two amino acids, and we call two amino acids joined together a dipeptide. If there are many amino acids linked in a chain, we call that a polypeptide, since poly means many. Now within an amino acid, we see certain structures repeating themselves for the 20 different amino acids. We always have a carbon atom in the middle, which is going to be bonded to four things. It's always bonded to a hydrogen, no matter what amino acid we're talking about. It also always has a carboxyl group sticking off to one side. It also always has an amino group, NH2, sticking off to one side. The only thing that changes between our 20 known amino acids is the R group. R is just a generic placeholder that could symbolize any molecule. And we'll look at a few examples of R groups that have particular properties. Okay, here it says there's 21 known amino acids used in biological systems. I think it's 20, but I'm not sure. I will check on that. It also tells us that all amino acids have that amino and carboxyl group, and we have that central carbon. They also talk a little bit about the, oh, hello. Oh, my computer doesn't like me right now. Cancel. Shoot. I think it's still recording. Oh, we're still going. Perfect. Okay. Sorry, I'm a total weirdo. They talk also about the R group and how there are a bunch of different types of R groups. We're going to look at several different types, which are in your notes packet. Within a protein that has multiple amino acids linked together, one end of the chain is always referred to as the N terminus. That's going to be the end of the polypeptide that ends with an amino group, the nitrogen. And we also have a C terminus that ends with the carboxyl group. So we say that protein molecules or polypeptides have directionality in that they have one N terminus and one C terminus on opposite sides of that molecule. so now we're going to focus in on a few different types of R groups. On your notes, I indicated where the central carbon is located. For this first one right here, they don't actually write in the carbon atom. There's a carbon right here, and there's also a carbon right here for the carboxyl group. This is your central carbon for this molecule, and they don't even show the hydrogen atom sticking off of it. But they do show a very, very large R group. It's got carbon, 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 and then it's got some nitrogens and hydrogens. The key thing about this one, arginine, this particular amino acid has a charge, a positive charge. This one also has a positive charge, lysine, and we have a couple examples, glutamic acid and aspartic acid, that have negative charges on their R groups. Okay. We also have amino acids that are not charged, but they do have polar R groups, in that regions of the R group have slightly positive charges versus slightly negative charges, but no overall charge. Both these polar and our charged R groups are going to be attracted to anything polar or charged. Here are special case R groups. Special case R groups include any R group that has a weirdo atom in it, like sulfur. This is called cysteine. It's the only amino acid that I know of that contains sulfur. And the sulfhydryl group at the end of the R group can actually bond to another R group that has a sulfhydryl group. So you're going to see amino acids across the protein bonding to each other if they both have sulfhydryl groups. That can cause the protein to fold, and if it changes its overall structure, it'll change its function. The only other one I want to mention here is glycine. Glycine, here's its central carbon. Its R group is actually just a hydrogen that's not pictured here. Okay. And then our last category are going to be our amino acids with hydrophobic or nonpolar side chains. Side chains is another term for R group. And you're going to see chains of carbons and hydrogens, but not much else. Whenever you see carbon and hydrogen, you should think to yourself, oh, I think that's nonpolar. It looks a lot like a fatty acid. We also have these guys right here. This is known as a benzene ring, and it's a bunch of carbons that are linked together, and they're held together in these places where we see the two lines by double covalent bonds. Okay. All right. I told you that proteins can get really complicated, and here's where they get super complicated. Because we have so many potential functions for proteins, we need to have a bunch of different possible shapes or structures. There are 4 levels of protein structure at which different proteins can vary or differ. The first level is called primary structure, and this is going to be the actual sequence or order of amino acids within the polypeptide chain. If we have 20 possible amino acid choices and we have a polypeptide that's 100 amino acids long, think about the many, many, many different combinations or orders that we can have of amino acids. The cell determines how to order those amino acids based on the order of nitrogen bases in the DNA molecule. So the code in DNA determines the amino acid sequence within proteins. Secondary structure is our next level. We're still looking at a single polypeptide here, a single chain of amino acids. And secondary structure involves 2 different, folding or shape patterns that we see as a result of hydrogen bonding between carboxyl groups and amino groups on amino acids that are not next to each other. So you see up here, they're showing our hydrogen bonding between two different amino acids, between our carboxyl and our amino groups. This is one conformation that can result from that hydrogen bonding. It's called a beta pleated sheet. It kind of looks like crimped hair, very 80s. I don't know if you guys will even know what that is. But anyway, alpha helix is like a spiral. That's another conformation that can form as a result of these dotted lines or hydrogen bonds. All proteins have similar secondary structures because they all have amino and carboxyl groups within each of their amino acids. So the alpha helix and beta pleated sheet are going to be seen within any protein. Tertiary structure is something that makes each protein very, very unique. And it is determined based on interactions between the R group for each amino acid. So if you'll see up here, we have a hydrogen bond forming between R groups on amino acids that are really far away. That causes our protein to fold. Here we see cross-linking between two sulfhydryl groups on R groups that are not adjacent or not neighboring each other. The types of interactions that can occur between R groups include hydrogen bonding, disulfide bridges or cross-linking of two sulfur atoms, hydrophobic interactions, so attraction between two nonpolar R groups, hydrophilic interactions, attraction between two polar R groups, or even ionic bonding, attraction between two totally charged R groups, like these ones right here. Now you're saying, wait, we saw hydrogen bonding back in secondary structure. What's the difference here? Well, here hydrogen bonding is not occurring between carboxyl and amino groups. It's occurring between R groups. Each protein has a very, very unique tertiary structure. So we say that the actual shape of the protein is pretty much determined by the tertiary structure. Up until this point, we've just been talking about interactions that can occur within a single polypeptide. Within our last level of protein structure, quaternary structure, we are looking through the lens of multiple polypeptides that are folded around each other to give you a full protein. An example of a protein that's made of multiple polypeptides is hemoglobin. It actually has two identical polypeptides called alpha hemoglobin and two identical polypeptides called beta hemoglobin. Together, those four polypeptides will fold around each other in unique ways to give us that hemoglobin protein. And that's protein structure. Because we have so many different levels where the structure of a protein can change, we have a lot of potential functions for proteins. And they're pretty much the workhorses of your cells. You don't have to memorize any of these. I'm just going to do a real brief overview of some example proteins. You have proteins that are used for storage. They mention a protein found in egg white that's used as storage energy or an amino acid storage for the growing baby chicken. Then we've got hormonal proteins. An example of a hormone that's a protein is insulin. Insulin is used to regulate blood sugar. Structural proteins, you've got keratin in your hair and nails. It's also found in cow hooves. Collagen is another protein that's found in connective tissues within your body, like tendons and ligaments. Defensive proteins include the antibodies. Antibodies are proteins that are made by your body in response to a specific virus or bacteria, and they can mark that virus or bacteria for destruction by other immune cells. We'll roll on over to the other side here. We've got transport proteins. There are some proteins that are located in the cell membrane, and they act as channels to allow large molecules through that cell membrane. We also have hemoglobin. Hemoglobin being a protein found in your red blood cells that binds to oxygen and helps carry it through your bloodstream. We have receptor proteins located on the surface of cells. An example might be receptor proteins on the surface of your muscle cells that receive signal molecules called neurotransmitters from your neurons in your nervous system. And those signal molecules, when they meet up with the receptor, will initiate a sequence of signals that causes that muscle cell to contract. Then you actually have proteins within your muscles that are responsible for movement. We've got actin right here, this long chain, and then myosin. Myosin has what are called heads, and they'll do kind of a, what is it called when you tug of war with the actin, where they will grab onto the actin and they'll pull, and that causes contraction of your muscles. The last and perhaps most important type of protein are enzymes. You have millions of different types of enzymes in your body, and enzymes help to speed up chemical reactions. If you didn't have enzymes, your metabolism would be too slow to keep you alive. Okay, so now I want to take a look at an example of how protein structure affects its overall function. Normally, the hemoglobin protein is shaped in such a way that it allows for a donut-shaped red blood cell, which is very effective at carrying oxygen through the bloodstream. However, there are certain people who have a mutation in their genes that causes a disease called sickle cell anemia or sickle cell disease. This is what hemoglobin normally looks like. It's got a nice place right in the middle for oxygen to bind. However, people who have sickle cell disease, their genes code for a defective hemoglobin protein that has one amino acid that's been switched for another. So the problem is they're switching the normal amino acid, glutamic acid, with an incorrect amino acid, valine. Valine is hydrophobic, whereas glutamic acid is hydrophilic. So they have very different properties, and they result in that protein folding in a completely different way. In normal hemoglobin, the molecules are free-floating. They don't interact with each other. But in sickle cell hemoglobin, those hemoglobin proteins tend to clump together. Problem with that is that results in the cell in which the hemoglobin resides, the red blood cell, being an abnormal shape, being a crescent moon shape as opposed to this nice donut shape up here. Crescent moon shapes are not effective at carrying oxygen, and they can also clog your arteries and your veins. So that could put someone at an increased risk for deep vein thrombosis, clots in their legs, or heart attack or stroke. Okay. There are a couple conditions or a couple things that could cause a protein to actually lose its shape. The two conditions that most often would cause a protein to lose its shape are drastic changes in pH, the acidity or basicity of the environment that protein is in, or a drastic increase in temperature. The process of a protein losing its 3D shape is called denaturation or denaturing, and it results in the protein completely losing its function. The problem with people getting a very high fever is when you reach around 104 to 105, you run the risk of starting to denature your brain's enzymes, and that could be a big issue. We have a bunch of computer programs now that can actually help scientists visualize the 3D structure of certain proteins. And there is a game, I can't remember what it's called, where they had people trying to determine protein shape by just messing around with it. And those people messing around with it helped the scientists with their research because they were able to see different options for protein folding. I don't know what this is. Who cares? So spider silk, type of protein. And now we're on to our nucleic acids. I'm going to take a break. I'm going to have a snack. I'll be right back. I love you guys. I don't know how I talked about proteins for 20 minutes. That's absolutely disgusting. However, we're going to continue now with the lovely nucleic acids. Uh-oh. It didn't want to work for me. Uh-oh. Oh no. We're going to have to start over. All right, we're good to go now. A little general information for you about nucleic acids. They are made out of carbon, hydrogen, and oxygen, just like all of our macromolecules. They have some nitrogen in them, and they have some phosphorus in them as well, which is an element that we have not seen up until this point. We've got two major kinds of nucleic acid polymers, DNA and RNA. And both of those polymers are made out of monomers called nucleotides. Nucleotides have three main parts to them. I want to zoom in on this little nucleotide right here. Each nucleotide within the larger polymer is made out of a sugar. It's a pentose sugar because it has 5 carbons to it. That's going to be bonded to a phosphate group. And those two molecules, the sugar and the phosphate group, make up the sides of the ladder. The molecules that we see in the middle of a DNA or an RNA molecule are the nitrogen bases. There are 4 nitrogen bases in DNA, A, C, T, and G. I am sure you've heard of those guys before. Okay. There are a few differences between our two nucleic acid polymers. The first has to do with the sugar that's located within their backbone. Deoxyribose is the pentose sugar used in DNA, and that's what gives DNA its full name, deoxyribonucleic acid, a nucleic acid that contains the sugar deoxyribose. RNA, or ribonucleic acid, contains the sugar ribose. With regard to their nitrogen bases, DNA has our A's, T's, G's, and C's. RNA has 3 of those bases, but it does have one different. Instead of having thymine, it has uracil, which does have a slightly different structure. Additionally, DNA looks like a spiral staircase. It has 2 chains or strands that are bonded to each other, and it looks like a ladder. RNA is just a single strand with its nitrogen bases hanging into the middle. The function of DNA is to store genetic information, and that information is heritable. It can be passed down to offspring. The information in DNA is later used to create protein molecules that serve a bunch of different functions within an organism. The basic structure of DNA is 2 chains of nucleotides that are covalently bonded together. And we saw how the sugar and the phosphate groups in that sugar phosphate backbone are covalently bonded to one another. Make you dizzy. Okay. Those covalent bonds that link two different nucleotides together are formed by dehydration synthesis, and they're called phosphodiester bonds. In contrast to the glycosidic bonds from carbohydrates, ester bonds from fats, and peptide bonds from proteins. Across the double helix, our nitrogen bases, here's an A and a T, are bonded together by hydrogen bonds, and you can see those hydrogen bonds as dotted lines here. Certain nitrogen bases always bond together across the double helix. A will always pair up with T, and G will always pair up with C. There's two types of nitrogen bases, purines versus pyrimidines. And purines, let's go back to the images. Purines are larger in structure. They have 2 rings to them, whereas pyrimidines only have a single ring. I always remember that pyrimidines has a longer name, but it's a smaller molecule. Purines has a shorter name, but it's a larger molecule. You always have to have a purine like adenine matching up with a pyrimidine like thymine to maintain the width of your double helix. You're always going to have 1, 2, 3 rings across that double helix. Okay. Now, we didn't just figure out DNA structure out of the blue. Watson and Crick are the scientists that usually get the most credit for determining the double helix structure of DNA. However, their discovery was totally contingent on the work of Rosalind Franklin. Rosalind Franklin used a technique called X-ray crystallography or X-ray diffraction to image a crystal of DNA. This is photo 51, the picture that she generated that Watson and Crick analyzed and were able to use to come up with their double helix structure. To me, it looks like total garbage. However, to them, the experienced eye, that looks like a double helix. Wilkins and Franklin, well, Wilkins was an associate at Franklin's. They did the actual grunt work, but Watson and Crick analyzed the data. Watson, Crick, and Wilkins did win the Nobel Prize, but Franklin unfortunately did not because she was already dead at the time. Bam, bam, bam. Actually, let's take a look at the technique of X-ray crystallography. You have your X-ray source with an X-ray beam, and you shoot it through the crystal that has DNA in it, and then based on the pattern that those X-rays make on a film, as they bounce off of that crystal, you're able to determine the structure of the molecule within the crystal. Okay, RNA or ribonucleic acid, it is not the master copy of genetic information. Instead, it's like a messenger molecule. It takes the message from DNA and takes it out to another part of the cell called the ribosome and tells the ribosome how to make certain proteins. We also think RNA was probably the first nucleic acid, the first genetic material, because we know of certain RNA molecules called ribozymes that are enzymes. They can help with their own replication. So they could copy themselves and pass themselves down to new cells. There are a lot of different types of RNA. 3 that we're going to look at throughout the year are messenger RNA, transfer RNA, and ribosomal RNA. Because RNA is single-stranded, it's a little bit less stable than DNA. Occasionally, base pairing will occur between an A, an adenine, and a U, a uracil, and that'll cause the RNA molecule to not be a straight chain, rather kind of folded. All right. So the basic pathway that information follows to get from DNA in the nucleus out to the ribosome to make a protein involves the use of RNA. We take our DNA in the nucleus, we make a copy of it, mRNA or messenger RNA, which gets sent out of the nucleus to another organelle or cell part called the ribosome, where it gets translated into a sequence of amino acids to make our polypeptide. Okay. At this point, I am going to let you know what you need to prepare for your DBA. I don't want you to worry so much about these. I'm going to give you my own list of things to study in preparation for your DBA. To show you guys how to prepare for your DBA, I'm going to show you the actual DBA. The first part of the DBA involves the functional groups. And I've got one chart where you're looking at an image of a molecule. This first one's cocaine, oh my goodness. And there will be an arrow pointing to a particular functional group. You'll have to write the letter from the term bank that corresponds to the correct functional group that's being identified in the space. Pretty simple if you know the functional groups. In our second chart, I will give you the name of the functional group, and you'll have to identify the name of molecules that contain this functional group, like alcohols for hydroxyl groups. In the third chart, I'll give you the name of the functional group, and you'll have to identify from the term bank the properties of molecules that contain those functional groups. Next, I'll have a series of statements that describe one of the macromolecules. These might be statements about the monomers, the polymers, or about the functions of that macromolecule. So you'll have to know the monomers, polymers, and functions for each. And then you have a section where you're shown a picture and asked to identify which macromolecule is being shown. You'll also have to indicate whether that image shows a monomer or a polymer. Okay. You'll have 6 multiple choice questions. What you'll want to study for those multiple choice questions is the difference between the 3 types of isomers. This is something that we talked about in class when I was there. There's 3 types. These molecules have the same molecular formulas like C3H8. However, they have different connections between the atoms within those molecules or different shapes. The 3 types of isomers are structural, cis-trans, which is also called geometric isomers, and enantiomers. You should be able to recognize that 2 molecules represent a particular type of isomer based on an image. There's an example of each type of isomer in your notes, so I would suggest looking at those. Also, you should be able to describe the difference between dehydration synthesis and hydrolysis and recognize examples of each. You should also know the names of the covalent bonds that are formed during dehydration synthesis and broken during hydrolysis for each of our 4 macromolecules. Those are the glycosidic bonds for carbs, ester bonds for lipids, peptide bonds for proteins, and phosphodiester bonds for nucleic acids. All right, guys, that sums up macromolecules. I hope you're not too terribly bored. Love you all dearly. I'm sad I wasn't able to be there. Okay, bye-bye. Oh, one more thing. Your homework is to finish the macromolecules worksheet that you're given in class today, and also to prepare for your DBA next class. The DBA is particularly long, and if you do not prepare for it, you're going to tank it. Very sad. We don't want that. Bye-bye.